Hi, my name is Tyra Countess, and this is my presentation on initial investigation of wildflower honey using headspace solid phase microextraction coupled with gas chromatography mass spectrometry for geographical information. So honey can be used in a variety of different ways ranging from food to medicine. It is thought that you can address seasonal allergies, especially after a move, by consuming local honey. While honey is mostly made up of sugar and water, there are other components that include minerals, phenolic compounds, organic acids, proteins, vitamins, and volatile compounds. These are usually referred to as volatile organic compounds. These compounds can include a variety of different chemicals, including esters, ethers, alcohols, carboxylic acids, aldehydes, ketones, terpenes, non-isopropanoids, carotenoid derivatives, furan and pyran derivatives, and phenolic volatiles. Basically, all of these components are specific to geographical locations. Because of this, each honey has a unique thing called an aroma profile. The presence of certain compounds can be used to verify the origin of each honey. Because of the difference in chemical makeup of each honey, it is thought that consuming honey made locally will help aid in treating seasonal allergies. This idea comes from the thought that the composition of the honey will build up tolerance to local allergens like pollen. In this experiment, we analyzed volatile and semi-volatile aroma compounds and a variety of different wildflower honey samples from different locations to detect regional variation in each honey. This was conducted by using headspace solid phase microextraction to extract aroma compounds from each honey sample, and then gas chromatography mass spectrometry to detect and isolate volatile organic compounds from each sample. Finally, cluster analysis was conducted to try to separate honey by region. In this experiment, we used 17 different honey samples that are shown in this table. These samples vary in location, showing that some are purchased from around the area of Conway and other parts of South Carolina, and then others are purchased outside of these in several different states, as well as one from Brazil. All the honeys are kept and sealed at room temperature until they're ready to be used. Each sample was kept in a headspace vial that was made using 5 grams of honey, 1 gram of sodium chloride, 5 milliliters of water, and 50 microliters of standardized solution to standardize all the samples. This is made of a 2-heptanol and glycol solution. These samples were then heated at 45 degrees Celsius for 15 minutes with stirring. The solid phase microextraction fiber was injected into the vial in the headspace above the liquid for 40 minutes. This is done while continuing stirring and kept at the same temperature. Once this is done, the fiber can be removed and injected into the gas chromatography injector for 2 minutes to be analyzed. We carried out this process 3 times per sample. So first we took a look at the compounds that were found in all 17 of our honey samples, and this includes butyryl benzaldehyde, octanyl, 2-methylbutanol, and pentadecane. Using average relative area, we compared the composition in both local and non-local samples. This data shows the differences that the different regions have when it comes to chemical composition of each compound. Next, we performed a principal component analysis on the data that was collected for each sample. This was done using average gas chromatogram peak height that was recorded using gas chromatography mass spectrometry. This result showed difficulty in forming conclusions and meaningful clustering. Several principal components are needed to explain most of the variation here. So from this, we were able to conclude that the gas chromatography mass spectrometry was successful in being able to detect and identify different chemical compounds that make up the wildflower honey headspace, and that the PCA clustering was able to differentiate each honey from one another. However, the PCA did not successfully cluster each honey by region. Even so, this works to show that the potential for using headspace solid phase microextraction coupled with gas chromatography mass spectrometry to identify regional chemical markers to establish location of each honey. In future experiments, we plan to add additional samples both locally and nationally to help better the geographical clustering, as well as using nuclear magnetic resonance or NMR to also help with clustering analysis.